oxysomes. They are found in chloroplast, mitochondria, and bacterial cell, and they are used to synthesize ATP molecules, the energy molecules. And they are known by different names also. They are known by elementary particles, primary particles, Fernandez and Morin particles, Parsons particles, F0, F1 particle, ATP synthase, coupling factor, and smallest rotating unit of the universe. Come to the structure of oxysome. And there are two distinct parts of oxysome, F0 particle and F1 particle. F0 particle is hydrophobic in nature and it consists of three types of polypeptide chain, A, B and C. There is single A polypeptide chain and 2B polypeptide chain and 10 to 14, 10 to 15 C polypeptide chain. And C polypeptide chain, they make the ring-like structure of oxysome. And now come to the F1 particle. F1 particle is hydrophilic in nature and it consists of 5 types of polypeptide chain: alpha, beta, gamma, delta and epsilon. And there are 3 alpha polypeptide chain, 3 beta polypeptide chain and one of each gamma, delta and epsilon polypeptide chain. Gamma and epsilon polypeptide chain, they make the stalk of oxysome. And alpha and beta, they make the hexamer structure. And in a hexamer structure, there is a central cavity, and in the central cavity, there is axis of gamma polypeptide chain. And in this hexamer, alpha and beta polypeptide chain, they are alternately arranged to each other. And this delta polypeptide chain, it makes the connection in between F1 particle and F0 particle through these two B polypeptide chain of F0 particle. And we can divide all parts under two group also of oxysome. One is rotating part and second is static part. Rotating part includes this C ring of F0 particle and gamma polypeptide chain of F1 particle, and all other parts, they are kept under the static part. And now come to the functioning of oxysome, how the ATP synthesized, how the ATP formation takes place. ATP formation takes place when there is gradient, when there is proton gradient is developed. Proton gradient means there is difference in the proton concentration in between matrix of chloroplast and mitochondria and intermembrane space of mitochondria and lumen of thylakoid. And there is greater proton concentration in intermembrane space of mitochondria and lumen of thylakoid greater than the matrix. And this proton gradient, it makes the movement of protons from this side to this side through A polypeptide chain and C ring. And by the movement of these protons, there is rotation of C ring and gamma polypeptide chain. And this rotation makes the conformational change, structural change in the beta polypeptide chain. Actually, beta polypeptide chain of F1, it works the main catalytic site of oxysome. And during conformational change, there are three stages of structural change. One is loose form. In loose form, ADP and inorganic phosphate, they come together. In tight form, formation of ATP takes place by come close together of ADP and inorganic phosphate. And in open form, the produced ATP is released into matrix.